is my slide?
That's all right, I can find it, don't worry. I'll load it. Right, good morning. We'll make a start. I'm Roy Goodacre. I'm convening this workshop. Nick Rattray, who basically put this together, is unfortunately not here. He's sort of cataclysmically done his back in and is waiting for surgery. So he was obviously told that he couldn't fly here. But fortunately, we have James McRae, who's going to stand in and give a presentation. So the objectives of this workshop were, first of all, to sort of highlight some of the history of clinical biomarker development, and in particular discuss some of the future trends and some of the statistics that might come along. So I'm basically just going to give a very brief overview of that, and then come in at the end, and hopefully we'll have some kind of organised discussion. So whilst people are talking, please think about questions that you can have at the end that are maybe more discussion-wise. If you've got specific focus questions for speakers on their presentations, then ask those. But if you've got ones that are more general, if we can hold those for the end, that would be great. Those of you who are not new to metabolomics will know that it's not a single discipline, that it's a highly multidisciplinary science, and with that comes complexities of data, complexities of data analysis as well. And quality, like in every area of science, and reproducibility is very, very important. Okay, so if we have a look at D cubed, if you like, we think about data quality and data analysis. There's this quite nice editorial, if you've not read it, it's only about a page and a half, by George Poster, that was published 6 years ago now, and it's called Bring on the Biomarkers. And this puts clinical translation, I think, into context, which is really quite nice. And at that time, there were 150,000 biomarkers described as being useful in a paper. In other words, someone had done some study, they'd found something that would allow them to differentiate between maybe a case control group. And that was published, and that went through peer review. And you can see that's that big, huge black splurge. And the tiny little dot at the bottom are the 100 that are clinically useful. So when we talk about biomarkers, and maybe when you're listening to people at this conference, when they say, I've discovered this marker for X or Y, just think about this and how valid that might be. And this is why, unfortunately, most findings are likely to be false. And our first speaker was quite vocal on this and published this nice paper in Metabolomics on statistical strategies to avoid false discovery. A paper that's been very well received, has had several hundred, a couple of hundred citations more. So if you've not read that, please also dip into that, because it's a very nice starting piece in terms of how statistics should be considered during metabolomics analyses. So our menu, if you like, is David will start off telling us about metabolic phenotyping and discovery. James will then talk about understanding mechanism, particularly metabolic flux and pathways and diseases, and how those can help lead to drug therapy. And then Kevin will tell us about clinical translation with large cohort target studies. And then those discussion questions, hopefully, we'll have at the end. So without further ado, Dave.
Okay, thanks, Roy. Thanks for promoting the paper, it's always nice. So I've been asked to talk a little bit about metabolic phenotyping. My background's in biostatistics, machine learning. So it's a data-driven science, so I won't be touching very much on the analytical methods, the method prep. We had a very good workshop yesterday about that. So I'm just going to go very, very quickly, because I think I've got about 50 slides to do in 20 minutes, so it will flip along a little bit. But I want to talk about just generally in principle what we're talking about in terms of metabolic phenotype, the workflows that are involved, a little bit on quality assurance, and then the majority of what I'm going to talk about is probably in the statistics. So it's always good to remind ourselves that what metabolomics is, and when we actually want to talk about the complete set of low molecular weight metabolites found in biological systems. I've focused on complete because often people will push targeted metabolomics as being metabolomics, and to a certain degree it's a bit of an oxymoron. James is going to give us a good talk later on, but it's more, for me, that's more about understanding the biology and possibly validating some of the stuff that we do in the discovery. The only way you could really go to having a targeted assay become metabolomics is if you run a hell of a lot of targeted assays. And then we get to somewhere where semi-targeted. Now, semi-targeted is great. I mean, I know people who do this. It's obviously the problem there is you have to have a lot of samples to run through all those methods, and it's going to take a lot of time, and it'll probably take you a lot of money. Now, there is somewhere in the middle, and I'm speaking for Thermo on Wednesday lunchtime, bit of a promotion, and I'll be talking a little bit about the idea of being somewhere between semi-targeted and untargeted. And of course, there's companies like Biomarker that are in that space as well. But when we're talking about discovery and metabolic phenotyping, we are really talking about untargeted metabolomics, where the idea is to measure as much as possible and get as good coverage of the metabolic space as we can. Now, obviously, there's many, many thousands of metabolites, lots of which we don't even know what they are, so we're never going to get full coverage, but with the technology that we have, we really can measure several thousand metabolites or features, or then we have a big discussion about metabolite identification, which we'll have later. Just to give a very quick comparison of the profiling versus the targeted is pretty much the same technology can be used. In untargeted, it's relative quantification rather than absolute quantification. In untargeted, we're looking at global rather than a focused list of metabolites. We do the identification after we've done the data acquisition for the untargeted, whereas for the targeted, we obviously know the metabolite IDs. And then, importantly, we have a lot of data processing to do untargeted, whereas with a targeted assay, it can be very simple because if you're just measuring a few metabolites and you just want to know whether one's gone up and down, then you can just do a t-test. Very much, I always push whenever I speak, because this is where we started back in the early 2000s, where we always talked about metabolomics being a hypothesis generation, and it really is narrowing down the search space so that people who are going to do more target analysis and do some flux analysis have somewhere targeted to look. So our role, or my role, or the phenotyping role, is to try and get an idea of what's going on in a kind of general way. And just to really emphasize this, with a targeted method, a huge amount of time and effort goes into doing the method development and validating and doing a calibration curve so that you have a very, very strong method. Data acquisition is data acquisition. It can be fast or slow, but the data analysis is really small. With untargeted, with the profiling, it really is flipped on its head, and there's a huge amount of work that has to go into the data analysis. Method developments are pretty much standardized. When I was developing these slides, just popped into my head that it reminded me of a slide that came up in one of Doug Kell's papers in 2007, which really does indicate the fact that when we're doing the profiling and phenotyping, we are really talking about systems biology, where we're trying to study the system at a whole, and we're doing a kind of inductive reasoning. It's very much dependent on the data analysis. Nice paper if you ever get a chance. Where phenotyping comes in from profiling, it's a kind of a new term that kind of came really out of the phenome centers. For me, it's more a conceptual thing. We're basically using the same technology, but what we really want to be thinking about is large-scale studies where we start using the metabolome to look at interactions between the genome, microbiome, exposome, and lifestyle, and really doing the kind of epidemiological level studies where we're talking about high thousands of numbers. The sort of studies that we started off when we were doing the HuSMET project, which Roy led. So let's move on to workflows. Now, as Roy pointed out, this really is an interdisciplinary field, and as such, there is actually more than one workflow. So we always like to think that there's just one that we go onto a workflow online, which will help us with the data analysis. But there's an overall biological workflow, if you like, where they go from the concept through the study design, recruiting patients, collecting the samples, biobanking, doing the quality assurance, doing the chemical analysis, doing the quality control, doing the pre-processing stats, and then publishing. Now, we had a whole workshop on this yesterday, so I don't have time to talk about it, but if you'd like, this is a good place to start on Nature Protocols paper, again, something that came out of the HughesMet project. Whereas for the chemometrics, cheminformatics, we really have to, and this is what generally people think of when they talk about the data workflow pipeline, is the idea of getting the raw data and turning it into lists of metabolites. And I'm not going to go through this in detail. Again, there's actually the parallel workshop, so if you're really interested in this, you probably won't be in the room. But I just wanted to flash it up because it is quite a complicated workflow, and it can take a huge amount of time, particularly when you're down in this area of doing the metabolite identification. There are some nice tools now. I have ThermoLab, so I use Compound Discoverer, but there's the Progenesis, there's XCMS Online, and various other Galaxy workflows as well. And of course, there's a biostatistics workflow, and this is where I live. So for me, this is about checking your study, checking your data quality. At every point in that workflow that I described earlier, there's some statistics, there's some data checking, there's some reporting. We do some cleaning, we do some stats, and then sometimes we even try and correct for some of the confounders that we might have accidentally or unavoidably introduced during the experimental design. We can start putting those into our models and start getting some really, trying to get the biology out of the noise. But of course it doesn't end there. Really what we want to be doing as metabolomics now, as we're moving forward, is we want to be integrating our metabolomics data with all the other omics that's out there. And this is a big task. There's people who are moving into this area. If you go onto XCMS online now, if you go on there now, you can go up and check out this XCMS job, which is online, and just click and flick through it. But they've done some quite interesting pathway analysis where you actually can load in protein lists and gene lists and map them to pathways, so that you can see here it's a kind of a cloud map of significance and percentage coverage. And you can see here that we've got a 78% overlap and a list of metabolites and genes and proteins that are all associated with that pathway. Now, this is done in a very, to my eyes as a statistician, quite a simplistic way, but it's a starting point. It's somewhere where we can start getting the grasp of the biology. Where I want to be, and this is where I started, I did my PhD in machine learning, I want to start looking at how we can actually mathematically pull these data together, and this is where we're going to be looking at using modeling techniques like ONPLS. And Johan Trieg is speaking tomorrow morning as a keynote. If you go and see anybody in this conference, go and see him. He's probably one of the most important people that have been involved in data science. He's probably one of the most important people in the metabolomics community, full stop. And hopefully he'll be giving us some insights into some of these more, these newer methods for trying to pull in, looking at genomic and metabolomic and transcriptomic data as well. So I was going to talk a little bit more extensively about quality assurance, but again, we had a fantastic workshop yesterday, and we had a fantastic lecture on this. So I'm just going to very, very quickly. We've written some papers. There's an issue with quality control, pooling samples so that you have some idea of getting how reproducible your data is. And we've presented work where we can look at the QCs through an analytical run, and if there's drift, we can correct for that drift within a batch, and we can also correct for drift between a batch. And then the idea being that we try and remove noise in the data. So from the left-hand side where you can see we've got a huge drift of QCs, and these numbers are all the same samples, just repeatedly analyzed. You can see after the QC correction and a bit of cleaning that we get quite a nice clusters that you would actually start believing that this PCA plot is real, whereas this one you wouldn't want to take any biological understanding from something as noisy as that. So, statistics. And this is a very, very important point, because we always think about biomarkers as being a list of analytes, and that's it. You will get a pattern. I'm on a pattern where the pattern basically lists 15 metabolites. But really, a metabolite, a biomarker, an omic biomarker, or a multifactorial biomarker is composed of the multiple molecular measurements as well as a fully specified computational model. And the fully specified, I didn't highlight it here because the computational model is the important fact, but it's the fact that you've got to have a very robust model if you're going to pull together 10, 15 metabolites into a single biomarker test, you have to make sure that it's a very robust model, because in order for it to be generally applicable to the whole population. And of course, we're dealing in a systems world. The whole point for me of doing untargeted metabolomics is we're measuring all these metabolites at once, and therefore we can look at the interaction. It seems so dumb to me that you would pull this data apart and do a list of t-tests and then just create a table of which metabolites have gone up and down when we've got all this data that we could start looking at the correlations between them. And one way we will do this is by using data projection. PCA is the very obvious one. And this is just a quick animation to give you an idea of what we're trying to do when we're doing this projection. We've got a three-dimensional space here. I know in metabolomics we're dealing with 150,000 dimensional space, but this is just a figure to give you an idea. We've got a noisy cloud, and the idea is we're going to rotate this data and project it onto the wall, as the previous slide suggested. And the idea is to rotate it until we get some information that's of interest, and that's when we stop the rotation. And that's the projection we're interested. And the amount of rotation will tell you which of those axes is important in terms of that. So let's just start. I should have started before I spoke. So you can see, as we move and we shift the axis around, that sooner or later, we do actually get something useful. Oh no, how do I stop it? There we go. So if you do PCA, so PCA looks at the maximum direction of variance in your data. So luckily in this data set, the maximum direction of variance happens to be this bear. Unfortunately, we can have very noisy data. And this time it really does take a little bit more effort to kind of get that rotation right and really to see what's going on. And again, we're really, this is looking, it's colored, so there's a little bit more information there that we might use to pull out. But again, if we rotate it at a certain angle, we get something that quite nice. But if we just done the principal components analysis, you can see that the statistical variance in the data is actually a lot of noise. So this noise could be instrument noise, it could be due to sample collection, it could have been variance in the way things are collected. So this is where we have to move to using linear discriminant analysis, and this is where we give it some of the information, and that's what the color coding was in the animation. If we tell it that this guy's hair exists, and they should be separate, and his face is a separate entity, and his bow tie is a separate entity, and then you look for directions in the data of maximum discrimination rather than maximum variance, then you can pull out information out of the noise, and this is very, very important. So again, metabolomics is multivariate. This is a paper that I think was published last year on gastric cancer, and this is a spring plot which shows the urine metabolic profile for the disease, case control, red being upregulated, blue being down, size of the spot, biggest, more important, and the springs are the correlations. So you create a spring between each of the points. If the strings relate to the correlation, then the tighter the string, the higher the correlation, and if the correlation isn't significant, the spring's cut. And then you relax it and you end up with these kind of nice clusters. It's a nice way to just get a quick visualization of what's going on. But really we want to pull that together into some sort of biomarker test. This is interesting for understanding the biology, but if you want to do a model, then you really want to start doing something a bit more clever. Now, the most popular method would be PLS-DA, and this is an example from that data set, 25 cases versus 25 controls. We've done the PLS, R squared of 0.73, Q squared of 0.62, and we've got a fantastic area under rock curve, which is over here. So this is showing you the performance of the model in terms of discriminating between the cases and controls. And down here, we've got an indication of how important each of the metabolites that were measured in this NMR profile, where the higher the peak, the more important. This data's been normalized, so this is most important, second most important, and then there's a few down the bottom that are important. Quickly flash this up, because not enough people do give information about how they've optimized the complexity. So this is an R squared Q squared plot, and this is where we decided the number of latent variables, which is 2, is optimal, because you can see the cross-validation starts falling apart. Just to flash back to yesterday, I gave a talk pretty much on sampling variance. If you were at if you were at that talk, this will probably make sense. If you weren't, this might not make any sense whatsoever. But the idea here is you've got a population of, say, 10,000 people that have got a disease. Let's call it Broadhurst disease. And when you do an experiment, you do a sample of that 10,000, and you will calculate a metric. In this case, we're calculating the mean of those samples. But what you can see there, that every time we repeat that experiment, we're sampling from a different random set of people, you get a different answer. So it turns out that if over 100 experiments, randomly selecting 10, you get quite a big variance in terms of those mean values. Fortunately, though, the means of the means are actually identical. So if you average those means here, that center point is exactly that center point indicated there. And also that the standard deviation of this space, of this cluster, is equal to the standard deviation of this of the whole population divided by the square root of n, which is the central limit theorem. Now, and this is what we get our 95% confidence intervals, which is a really sensible way of determining whether you have a significant effect or not, far more useful than doing a p-value. And of course, the more samples that you take, the more samples that you select from that 10,000, the more accurate a single experiment will be, or more likely that it will be the true value. So if you get right up to doing an experiment where you happen to have got the samples from all 10,000, then you will definitely get the right mean. But obviously, when you're only getting 10, the variation is huge. So this really does give an indication of when you're doing sample sizes, it's very, very important to take this into consideration. Again, I can't go over the whole of the talk from yesterday. One thing I didn't talk about yesterday was bootstrap resampling, and this is the idea of trying to simulate that confidence interval by repeating an experiment hundreds of times. Obviously, we don't have the time or money to do that in the real world. So what you can do from a single experiment, in this case 50 samples, you can resample from your 50 that you've done, calculate the mean, and keep resampling from the same 50. And it actually gives a very, very good estimate of the true size. So again, I'm going to run this. On the left-hand side, it's very similar to the previous slide. This time we've got an N of 50, so you can see that the cluster is a lot tighter. But you can see here, we're subsampling, and you can see the cluster is actually quite similar. So this is the idea of bootstrap. Bootstrap means you sample with replacement. So you sample 50 from 50, but with replacement means you can pick some of them twice. So you're generally picking 2/3 of the data set as you go. Now, I needed to show those videos because what I wanted to show next was applying those principles to determine how well your loading plots work. So the idea here is we've got 25 versus 25, and we're going to do bootstrap resampling to get some idea of how sensitive these loadings are to the data. So we run this, and you can see that it's selecting, randomly selecting. And this is the discrimination. This is the PLS in effect. This is the different ROC curves for each of the models. So we're getting a cluster of different ROC models. And this is the variation that you're getting in your loadings. And it will go up to 100, so I'm going to have to keep talking. I'll have a drink of water. But just a point of interest, if anybody's interested in OPLS rather than PLS, this projector here, this is basically what OPLS is doing, is projecting it onto that plane so that the first component is the discriminatory component in terms of the biomarker, and the second component is the noise. It's doing that all in one. This is just, PLS is a lot easier to program. So we're getting close to, so there we have it. And look what we get. So the original loadings, when we used all the data are the blue spots, but just look to see how much variance there is there. And you might just be able to say whether something's significant or not, because basically if the 95% confidence interval, if that distribution is above the line or below the line, then you could say that metabolite is significant. But if you were going to use this as a biomarker test, would you trust those values that you created using 25 samples? I think not. It probably wouldn't work when you find another 25 samples from somewhere else in the world, throw it into that model. It's a very unstable model. So what I did, oh no, sorry. So, just for a bit of fun, I calculated the multinormal distributions that were in that data set. So I got the probability distributions, and then I resampled at a lot higher rate. So this is an artificial data set, and this is not what you would do. This is not something you would do for your data. This is just to illustrate the point of having a larger sample size. So in this case, it's exactly the same structure in the data. We're getting a very, very similar model when we use all the data, but this time we're measuring 250 versus 250. And you can see, PLS model, same kind of idea, big peaks are important, ROC curves very good discrimination. But now you see when we run this, I'm going to run it a lot faster, that you can see that there is variation in those loadings, but there is a lot less because you've got a lot more data to get the proper structure, latent structure that's behind the biology by having a larger sample size. But you can see it's a lot better. It's not perfect. So if we then do the final one, it keeps doing that, sorry. So this time, we've got 1,000 versus 1,000. This is more like it. And you can see how little the PLS projection is moving. This is a very, very stable model. There's one that's freaking out, so this is something you would probably want to remove before you go anywhere further. But look how good these values are now. So this is something, if I saw this, and I saw these values, then I would start believing that this was a true biomarker. And remember, the biomarker is the analytes plus the model. But you only get this quality of model when you're looking at thousands of samples. And this is, sorry. Figure out how to use Windows. So that's kind of where I wanted to end. So it was just some conclusion. Biomarker specification requires large sample sizes. Large cohort studies are required to do high quality data, SOPs. But the big thing is to move forward in clinical metabolomics, certainly at the discovery level, we really have to start doing very large data sets. And this was kind of a lot of the philosophy around the phenome sensors that are being set up around the world. So on that, I think I've almost on time. Thank you. Well, the idea is, when you do an experiment, you cannot sample the whole population of a disease group. Say there's 10 million people with a certain cancer. You do a subset to find your biomarker, yes? You understand that?
Yeah.
So, let me finish. So, when you do repeated, so you do that once, you'll get a single value. Now, if you do that again and you've picked a very small sample size, you might randomly find a subset of that population which is not representative. You'll get a completely different value. And then you do it again, you get a completely different value. Well, if you do that in terms of a case and control, imagine you've got that happening in two directions. So your control means over here, your disease means over here, one experiment, but then they're here for another experiment, and they keep moving around. You have to be very careful about how you power your study, and about effect size, about variance. Again, I spoke about this a lot yesterday. I can talk to you afterwards. The slides from yesterday, which were more about experimental design and sample size, are going to be available online through the society, but I'm happy to discuss that offline, but I think it's a bit too much to go into right now. I'm happy to talk to you and explain if you like.
Thank you. Thanks, Dave. Next up is James.
All right, yeah, so it's not all going to be about clinical biomarkers today. I was asked to step in at the last minute to replace Nick, so I'm going to talk really about what I still regard as metabolomics, no matter what other people say. But my first couple of slides will be metabolomics based, and it's actually relates quite well to David's talk. It's going to be talking about the importance of experimental design, and also sometimes, if David's going to have a dig at me, I'll have a dig at him. Having thousands of samples is lovely, but it's not really the real world that we live in. Most of the time, we've got very small numbers of samples, really, in terms of statistical relevance, I would say. So we have to work with what we've got. That's one of the challenges in doing any kind of metabolomics study, especially when you're looking at clinical cohorts, especially when you're looking at diseases that are very small percentages of the population. And my first 2 slides are going to be telling you about things that I can't tell you about. So there's a lot of, as Trump would say, fake news in this. Maybe I'm giving this talk a year too early. I'm not allowed to tell you anything. Even my picture of a kidney is incorrect. This is what I do is nothing to do with the kidney, so I had to pick a random organ. But what I am doing is looking at some cancer metabolomics. We're looking at a particular organ that is not the kidney. That is, by the way, drawn in turbulence over Oman, so forgive me for it being terrible. Anyway, as David's been talking about, there's a number of considerations. One is experimental design. One is the numbers that we've got, especially for controls. It's actually sometimes a lot easier to get the disease samples than the control samples. How those samples are handled. So again, David alluded to using samples in different places in the world. Are the people who are collecting those samples going to handle them in the right way, in the same way? And the reason why they maybe don't is because metabolomics is often a distant second in the priority list for what is happening to these patients. And overall, again, one thing that David will be pleased about is, can we really get any significant results from anything that we do? So here is an example of the experimental design that isn't necessarily what we've chosen, but it's been thrust upon us. So in this case, we've got actually 550 patients with 2 different kinds of cancer in a random organ. And we've got plasma and red blood cell samples from diagnosis, from their first surgery where the tumor is removed, this intra-biopsy period where we've still got plasma and blood cell, and then at the point of remission, after therapy where you may be having chemo or radiotherapy, and then finally at second remission. And second remission often means death. What this actually means is that, and that's dropped off the bottom, if obtainable at all. We can usually get quite a lot of biopsies from these last samples. However, at this point, the patient, unfortunately, is no longer alive, and you're left to the family of the patient to see whether this organ and any other material is still donatable to science, and this can actually really be a problem. So along this course, we've got plenty of samples in terms of plasma and red blood cell, and we generally have 1 biopsy at each point, and that one tiny piece of tissue isn't always enough to get great metabolomics data from. So this is just highlighting the challenges of that. Another thing that we've been doing, again, I can't tell you anything about it, again, it's not the kidney, but is looking at in vitro models of the same kind of cancer. And here we're doing, beginning some forays into mass spec imaging in partnership with the National Physical Laboratory in Teddington in London, where we're doing a number of different kinds of mass spec imaging. Again, if you're in the other session that David told you about, you'll be learning about mass spec imaging right now from Berin Boughton, but so I'm not going to talk too much about that. That's really his expertise and the expertise of others. And mass spec imaging is one of these newish compared to what many of us do, trends in mass spectrometry metabolomics, where we're getting 2D, really good 2D and sometimes 3D depictions of the metabolites in the tissue sample. There are problems there, of course. How do you get the tissue? How do you prepare the tissue? Fragmentation during preparation of tissue is a real problem, so there's a lot of work to be done there. However, one of the other things that people are doing, and again, this is really nothing to do with me, but I thought I would try and get some kind of clinical stuff into a non-clinical talk, and really at the cutting edge, excusing the awful pun in this case, we've got these new trends that have been developed in partnership with Waters and Imperial, things like the iKnife technology, where the surgeon can actually, while they're cutting into the tumorous tissue, they burn the tissue so that smoke travels up this knife and goes straight into the mass spectrometer. And you can look for these biomarkers or evidence of tumor tissue. And here at this point, in this beautiful, now over Saudi Arabia drawn tumor, you can see that there would be peaks that would be indicative of the tumor, and as the knife moves away into the healthy tissue, those peaks would disappear, and you could try to, therefore, delineate where the tumor is and stop the surgery. So these things are, again, they're in the infant stages, but this is really where the future is going. All right, so now that's talking mainly about the clinical work. A lot of what most of us do in the metabolomics field is still doing traditional metabolomics, and by that I mean going back to here and with these sort of tissue samples or with these biofluid samples, doing your basic, usually organic extractions and then running them through chromatography and mass spec. Here's an example of a GCMS chromatogram, and doing all of these beautiful multivariate analyses that David was talking about. But what you can also do that is really quite powerful is stable isotope labeling, and that's really a lot of what I do. And stable isotope labeling can really bring those biomarkers or those abundances or those differences or those significant differences home as to what they really mean. It can also highlight things that aren't significantly different when you just do simple metabolomics. So you can't just do your untargeted profiling. A good example of that is in the TCA cycle. Often you could have big changes in the TCA cycle but see no changes in abundance. You're getting differences in turnover, but the abundances stay the same. And in those sort of examples, you need stable isotope labeling. So what do I mean by that? It's when you've got a carbon-13 or a nitrogen-14 or various stable isotopes, and you use them as a carbon source or a nitrogen source, feed them into your in vitro system, or in vivo. You can do these kind of studies in mice and other organisms as well, and you can track where that carbon-13, in the case of glucose, is going. You can see, well, here it isn't going into inositol metabolism, or it's going down here all the way through here, and eventually ending up in fatty acid biosynthesis. And you can do that with glutamine or acetate or fatty acids themselves, and really track what's going on with metabolism. How do we do it? Well, we need to get into the spectrum of each metabolite, and really what you need to know already is what each spectrum is. You need to have the identified metabolite. And this is kind of my beginning of a crash course in how to do stable isotope labeling studies. So in this case, again, a lot of what I do is GCMS based, so all I'm going to show you from now on actually is going to be GCMS. And here is inositol. This is myo-inositol. This is the characteristic fingerprint for that molecule. You'll notice that I've written, instead of OH groups up here, it's OTMS. Anybody familiar with GCMS will know that you have to do derivatization procedures. And here, TMS is this trimethylsilyl group that we put on just to make each molecule volatile so that it comes off the GC chromatogram and we can measure it in the mass spec. So that's why I've written TMS here. But anyway, the point is that this is the characteristic fingerprint of inositol, okay? And these fragments I've shown with their various structures. This one here has got 4 carbons, 1, 2, 3, 4. This one's got 3, this one's got 3, and this one's got 2. Okay, fine. What we can do, though, is when we're looking at carbon-13 inositol, so this is where that glucose, carbon-13 glucose has gone in, it's gone down inositol metabolism, and the carbon-13 has entered that, then you could see that in this case, these carbon-12s are now carbon-13. This one's got 3, this one's got 4, etc. And lo and behold, the ion will change its in size from 305 to 308, 318 to 322. etc. And looking at this ratio is a real crude way of looking at how much label has gone through into that metabolite, and when you have that in partnership with all the surrounding metabolites, you can really get a view of how much flux or how much carbon-13 is going down each pathway, what is active at each different point. But it's not quite as simple as that. If we go back to this, you can see that there are these other ions here. For those of you in the LCMS world, this might look huge. In GCMS, these natural isotopes are really quite significant, and this is largely because of this silicon here. While carbon has got around 1% natural carbon-13, silicon has actually got 5% plus 1, and 5% plus 2. So it really becomes a big issue for us in the GC world. So the first step of what we need to do is get rid of those natural isotopes. So here's an example of an unlabeled fragment, and we've still got these very significant +1, +2, +3, +4, and we have to put these through an algorithm so that we can reduce them to 0 and get the true abundance of the M0. That's really important because when we're looking at the labeling, we need to work out what is just the natural isotope and what is real. So by doing this background subtraction, we can get a nice clean depiction of that there is 50% of the M0 and 50% of the +4 in this case. So now we know that there's a 50% labeling of this particular compound. But it's not quite as simple as that. So in this case, we've got citrate. Citrate's got 6 carbons. And in this little cartoon, we've got this ion here, which is the M0. It's the 375 M over Z. And here we've got the +6. And in this case, this is where all 6 carbons in this citrate are labeled, just like we see here. So this is about 25% of the height of this. So we can say that 75% of these molecules are labeled. However, what about this case? Here, we've got 25% unlabeled again, and here 75% have only got 2 carbons labeled. It's still 75% labeled molecules. Here, we've got a bit of a mixture, so we've got that fully labeled version here, and just the 2 carbons labeled, and the 4 carbons labeled, and they're all equimolar. So again, 75% of the carbons are labeled. If you looked at how much 75% of the molecules are labeled, but if you look at how much carbon's labeled, you can see here, overall 75% of these carbons are labeled, here only 25%, and here 50%. What's really telling us the most information, actually, is looking at the pattern of the isotopolog. So here you can see it's all the +6, all the +2, and here we've got a mixture. And this can really tell us a lot about the nature of each metabolic pathway. That might be a little bit confusing right now, but hopefully it will make sense in a slide or two's time. And to demonstrate that, I'm going to tell you very quickly, about how we can use these techniques to look at ways of trying to develop drug therapies towards a disease. In this case, my background is in tropical disease, and this is the life cycle of the malaria parasite, Plasmodium falciparum. I've got no time to tell you about any facts or figures about how deadly that disease is, but I think most people have heard of it. And the disease itself is caused by these blood stages here. This is the red blood cell stage. This is the asexual life cycle. This is what I'm going to focus on for the next couple of minutes. And then in order to pass into the mosquito and then be transmitted to the next mammalian host, they have to go through this sexual stage, this gamete form here, the gametocyte production. So just bear those 2 stages in mind for now. So I've already talked about the TCA cycle. It's a little bit of a favorite topic of mine. The Plasmodium TCA cycle, well, it wasn't supposed to exist. It wasn't supposed to exist because this enzyme here, the pyruvate dehydrogenase, although it does exist in the malaria parasite, it's sequestered over in this strange organelle called the apicoplast, which is a bit like a chloroplast that doesn't do photosynthesis. And its only purpose is to make fatty acids. That I'm not going to talk about, but what I'm trying to say is that there is no pyruvate dehydrogenase in the mitochondrion. So for many, many years, it was thought that there is no TCA going on at all in the Plasmodium parasite, despite the fact that all of the enzymes were there, apart from that one. So we do a little bit of labeling, and this is the crude labeling. So this is just looking at the total molecules that are labeled in each metabolite. Here's my red blood cell control. These are uninfected, and you can see that the black here means that no label is getting into the TCA metabolites, and some of them we couldn't see at all, which is not surprising because there is no TCA activity in red blood cells. However, doing this asexual labeling, where we're using either glucose or glutamine, we can see that with glucose, it's not a lot of label, but we are getting some label incorporation here, around the 20% mark. And when we label with glutamine, we can see quite a lot. However, bear in mind that this is over 48 hours of labeling, so it's taken a lot of growth, an entire life cycle actually for these parasites to get label incorporation into those TCA metabolites. So we knew that there was some TCA activity going on, but it really took the isotopolog analysis to show us what really was going on. Here's a cartoon to depict that. Here we've got our carbon-13 glucose. Again, glucose, 6 carbons. It's got 13 C at every position here. By the time it goes through glycolysis, it forms PEP, which has got 3 carbons, and pyruvate, which has also got 3 carbons. And that can then go into the TCA cycle. So acetyl-CoA, 2, and then this picks up with oxaloacetate to form citrate. So you've got citrate with 2 carbons, and then it goes all the way through. So all of these things are going to have 2 carbons. And of course, as you can imagine, when it goes around the TCA again, this citrate will pick up another acetyl-CoA, so it will now have 4 carbons. And essentially, the longer this goes on, you'll get more and more carbon getting into the citrate, so you'll have a pattern of +2, +2, +2. And that's exactly what we see. You see a lot of the +2 there, +2 there, +2 there. And these other TCA metabolites, which have only got 4 carbons, you see this characteristic +2, +4 pattern here. But we also see +3, +3, +3, and in the case of citrate, a bit of +5. And that's because this PEP can also be carboxylated to oxaloacetate, which can then be converted to malate in the cytosol. Then that can be transferred into the mitochondrion and be incorporated. So that +3 oxaloacetate can pick up a +2, and then it can become +5. So already, just by doing one experiment, we can see a lot of what's going on here. We can see that there is a classic oxidative TCA cycle. We can see that there are contributions through PEP carboxylation just from one experiment. We can also do another experiment with glutamine and see that the same thing happens there. So this glutamine is converted to glutamate, that's converted to alpha-ketoglutarate, and that can get in. In this case, it will pick up a cold acetyl-CoA. We're not doing glucose labeling here. So you'll see a citrate with +4, and that's exactly what we see, this +4 here. And then when it goes through again, it's a little bit more complicated in this case. You'll see some of this replaced. There are interconversions that go on down here in this sort of area. So you can see more and more +2 coming. So where more of the labeled carbons have now been replaced by the unlabeled acetyl-CoA carbons. Won't explain that right now, but essentially kind of the opposite to what we saw before. The longer it goes on, you might actually see some more +2. All right? So again, in a couple of experiments, we've shown that there is a canonical TCA cycle in the Plasmodium parasite, despite the fact that it wasn't believed to be the case. In the sexual stages, however, and I'll go through this really quite quickly, you can see that something strange is happening. Not only is the glucose labeling much more significant now in the TCA metabolites, but the glutamine is reduced in labeling, and also it's a lot quicker to get labeled. This is only a 4-hour label, and if we look at the isotopologs again, in this case in a time course pattern, you can see that, let's start looking at the succinate, just because it's a bit easier to look at. Here, this is the +4 isotopolog, so this is the maximally labeled version of succinate, and you can see that its maximum incorporation is happening very early, almost complete after 2 hours, and by 4 hours it reaches the top. Similarly, let's go up to the citrate now. You can see that the +6 version is really quite high, or as soon as about 2 hours. This is very different to the 48-hour labeling of the asexual stage. You can also see that the +5 is very high as well, which is showing that PEP carboxylation is happening a lot here too. So this all highlighted that specifically in the sexual stages that the TCA might be important, especially if it's going on so rapidly or the label is incorporated so rapidly. And I'm not going to tell you about the biological validation of that, but suffice to say lots of people have looked at TCA interference, knocking out or chemically knocking out various enzymes thereof. And what we can see is that in many cases, looking at the black lines is where we have biological evidence. You can see that knocking out or chemically knocking out each of these TCA-related enzymes we get a halt of progression into the sexual stages, almost completely, in fact. For some of the enzymes, we're seeing a stop even at the asexual stages, but again, I don't have time to go into that. The bottom line, really, is I can sort of talk about these conclusions that are the biological conclusions of what we've seen, that the glucose and glutamine feed into a canonical TCA cycle, that there's a switch in carbon source preference between the sexual and asexual stages, and that by blocking the TCA, we can lead to a halt in gametocyte maturation and therefore a halt in the transmission of the disease. What we really care about in this particular talk is the fact that stable isotope labeling has really been able to show which biochemical pathways in the parasite are the most important ones, and has allowed us to target specific areas of that metabolism for potential drug therapy in the future. I think that is definitely my time gone, so I'm going to skip over the next two slides and just finish off with my acknowledgements. Most of this work was actually done back in Melbourne, where I was before I moved to the Crick in 2013. All I'll do very quickly, I'll leave it at the previous slide, a guy called Volker Behrens in Jake Bundy's group back in 2011 wrote this program called Gavin3. It's a MATLAB-based program that really has saved me a hell of a lot of time in doing all of these analyses. Doing the, getting the abundances of each labeled metabolite is or each labeled ion is incredibly challenging, and this particular program has allowed me to be able to look at all of my samples, somewhat less than 1,000, and be able to see at a glance which are the labeled and which are the unlabeled metabolites. If I had an hour talk, I would tell you all about that. I'm sure you'd be very interested. But yeah, if you check out his publication in 2011, that will lead you to a great world of stable isotope analysis. Thank you for your attention.
All right, so my name's Kevin. At the Metabolomics Laboratory at the Baker Institute. I've been in the same group for about 6 years now. Started off as an RA and then ended up doing a PhD there. So even though we're called a Metabolomics Laboratory, we actually do focus quite extensively on lipidomics. So a lot of the results I'll be presenting today will be based off a lipidomics study. So we've got two strategies in terms of human translation. One is hypothesis-driven, basically where we identify a biomarker, identify some change in human profiling, and we validate these results in either cell models or animal models, and hopefully move forward towards clinical trials in terms of lipid modulation. But for today, I'll be giving a talk mostly on biomarker discovery, basically identifying changes in the human lipidome, and then validating it in an independent cohort, and hopefully move on towards prospective trials. So when people think about lipids, they typically think about clinical lipids, so HDL cholesterol or LDL cholesterol. But the lipids I'm talking about are these small molecules that make up these lipoproteins. So in this case, making the core of these lipoproteins are cholesterol and other neutral lipids like triglycerides and diacylglycerides. On the surface, we've got phospholipids, such as phosphatidylcholine here. And we also have other sphingolipids like ceramides, glycosphingolipids, and sphingomyelin, as well as other exotic species, such as modified lipids, whether they're oxidized or glycated, lysolipids, and free fatty acids. So in total, there's probably 10,000 different lipid species in humans, but most would be abundant at some level in plasma, about 1,000 of them. So to measure these compounds, we use quite a traditional method. So we use targeted LC-MS. So this instrument's the backbone of our laboratory. One advantage of working at the Baker Institute is that we're the only group that uses this instrument. This allows us to book, basically use the instrument for months at a time without stopping. So I'm going to be presenting results from one of the trials we've profiled. We started this trial about 4 years ago. This actual study was done 2 decades ago. So it was a study examining the effect of statin treatment in ischemic heart disease. So this cohort had about 9,000 samples. Basically, it was a double-blind placebo-controlled study. So for baseline samples, there was about 6,000 samples. And after a year, they were recruited back, and they had their plasma samples taken again. So a couple of the main outcomes for this study was cardiovascular events and cardiovascular death. So we had about 1,300 cardiovascular events in the study, and in the baseline samples, about 4,600 had no events in any of the follow-up time points. I guess the main takeaway point for this slide is that your clinical cholesterols are not very good discriminators between those who don't have events and those who do have events. So our hypothesis for this was that baseline plasma lipid species are associated with incident cardiovascular events or death, independent of traditional risk factors, and that plasma lipid species would provide improved prediction of cardiovascular events or death. So we had 3 aims: basically perform lipidomic studies on all the baseline and follow-up samples, plus QCs totaling about 12,000 samples from the lipid study. So we wanted to identify species that were associated with future cardiovascular events and also develop multivariate models to predict the risk of future cardiovascular events. So firstly, I'll go through the first aim. So we had to modify extensively our methodology. So we couldn't really do a false extraction for 12,000 samples. So the extraction itself was quite simple. So a previous PhD student worked on this. So it's a single-phase extraction using butanol methanol mixed with our internal Stansted. Basically, for every 10 microliters of plasma per sample, we spiked in 100 microliters of the solvent, sonicated for about an hour, centrifuged to precipitate the protein, and then transferred the supernatant into vials. So we can comfortably get about 486 samples done a day. It takes about 4 hours. So if you were in a rush, I guess we can expand this to about 8 hours. And the samples are then subsequently stored at -80 degrees. So we also included several QCs in our extraction. So for every 20 samples, we would include a plasma quality control sample. Basically, this is just a sample that's pulled from healthy individuals from the group. And we use this to monitor the extraction and LCMS quality. And then every 40 samples, we had a blank as well. So for the LCMS, I won't go into too much detail in this particular method. I'm doing a talk tomorrow, actually, on the updated version of this method, so we don't use this one in particular anymore. It took about 10 minutes per sample using a solvent gradient of THF and methanol. So one of the issues we had with this method was that THF is not very HPLC friendly. So in total, we had about 354 lipid species we could measure quite well. So for the actual LCMS run, we used 2 extraction batches. So at 10 minutes per sample, it took about a week to run 1,000 samples. And we repeated this about 13 times. So we also included a separate QC. So we termed this technical quality control. So they're quite similar to the pooled plasma controls, with the exceptions that the extracts are pulled together and split into 1,000 vials, which we stored away. And every day, we would take 1 vial out for repeated injections. And this is purely to monitor for LCMS quality. And this is actually quite invaluable when we were trying to troubleshoot some of the issues we had. So the intra-batch variation was quite good. So about 80 to 90% of the lipids we measure had a CV under 15%. Of course, the technical QCs always had a lower CV relative to the plasma QCs. So this is a cumulative CV plot, basically the X amount of lipids below a certain CV threshold. So we usually see a difference about 2% in terms of variation between technical controls and plasma controls. So while intra-batch variation is actually quite good, inter-batch variation is not as good. So we had to align our data using some sort of alignment. So this plot here is for one particular analyte, a plasmalogen species. So the black dots are the individual samples, the red dots are the median and the IQR, if you can see the error bars of those particular samples for each batch, and the green are the median IQR for the quality control samples. So we aligned this data by using the median values of the quality control samples. We generated response factor, which we applied to each batch. So basically each batch had the same amount of variation, except that now inter-batch, the CV is quite similar to the intra-batch variation. So the first thing we examined was trying to identify lipid species that were associated with future cardiovascular events. And to do this, we used a univariate analysis, so treating each lipid independently. We adjusted for 22 different covariates, so we had enough samples that this wouldn't be too horrible for it, I guess. So we used the Cox regression. We had time to event data for those events. We calculated hazard ratios, and then we subsequently corrected the analysis for multiple comparison using Benjamini-Hochberg. So looking at just the total lipid classes, you don't actually see much going on. So here we can see this is a forest plot, so the hazard ratios is on the x-axis and p-values, the corrected p-values are on the right-hand side. We only see two classes being associated with cardiovascular death, so phosphatidylethanolamine and phosphatidylserine. But if you look closely at the individual species, there were 55 lipid species associated with cardiovascular events and 95 species associated with cardiovascular death. So we plotted everything out. So the forest plot here is on the side now, hazard ratios is on the y-axis. So you can see here that for the orange dots, these are the corrected p-values, and the grey dots are basically every other lipid we measured, so there's about 354 here. You can see that only phosphatidylethanolamine was associated positively with cardiovascular death, and all the other classes tend to have a mix, basically some positive, some negative. So when we look at the top 20 lowest p-values, basically the ones that we believe were the strongest in association, you can start seeing some sort of patterns going through. So for example, negatively associated with cardiovascular death were polyunsaturated species. So in this current method, we don't actually know exact composition for most of these species. We do have some idea, but the updated version has a better indication of what they are. So most of these are actually omega-6 fatty acids. Another thing we saw were sphingolipids. Of particular interest were the subclasses. So we noticed that only those species with 16-0 or 18-0 and acyl chain for these sphingolipids were associated with cardiovascular death. This was particularly interesting to us because we're looking at ceramide synthases. Basically, two ceramide synthases were responsible for generating the 18-0 and the 16-0 sphingolipids. And this effect was really independent of the other enzymes responsible for the more complex sphingolipids. So the second hypothesis was that plasma lipids would provide improved prediction of cardiovascular events or death. So we developed a multivariate model to predict the risk of future cardiovascular disease. So firstly, we actually minimized the number of collinear lipids. So we had a correlation minimization technique. So this reduced the number of lipids to about 144. So we created a base model, a Cox regression based model with the significant covariates, so all 22. And then we did a feature selection based on AIC within a cross-validated framework, 5/200. And then we ranked the top 20 lipids in order. And then these top 20 lipids are then sequentially added to the base model to develop the actual multivariate model. And we determined the C statistics and the NRI, both categorical and continuous, for relative to the base model with confidence intervals. So for cardiovascular events, the base model had a C statistic of about 0.645, and the base model for cardiovascular death had about 0.7. So incorporation of lipids improved the models independent of the 22 covariates. So we typically don't use all 20 lipids in modeling. So there's a point where adding more lipids doesn't necessarily provide much more information. So here we decided to pick 8. On the left is the cardiovascular events and the lipids we use in that order. And on the right is cardiovascular death and those lipids used in that order. So the red highlighted lipids are the species that were overlapped between the two models. So for cardiovascular events, we improved the C statistic about 22.1% for 8 lipids. The categorical NRI was improved by 8.2%, and the continuous NRI was improved about 30%. And for cardiovascular death, we improved the model about 3.8%, NRI of 10.3 for continuous, and 42% for, 10% for categorical, and 42% for continuous. So just to sum up, so plasma lipids were associated with future cardiovascular death in secondary prevention. So these were independent of traditional risk factors, and it provided some biological insight. And plasma lipids can predict cardiovascular event and death, so it actually improved upon traditional risk factors. And for translation, it's unlikely we'd do each individual species as is. We will likely involve simple lipid ratios in terms of modeling. So I'd just like to finish off by talking about some success. It wasn't our lab that did this, but in the Mayo Clinic, they actually launched a blood test in the US that assesses the risk of heart attack using plasma ceramides. So they found the same thing. 16-0 and 18-0 were quite associated with cardiovascular events. So they validated this result in a clinical trial of 4,000 subjects, and they ended up using ceramide ratios to generate a risk score to predict your chances of a heart attack. And that's it. So I'd like to thank everybody for everything.
I'll just have a few slides that I'll use for the discussion, and then maybe we can get the speakers to sit on the stage. So what we've basically heard today, I think, is that, as is always the case, experimental design is important. Numbers are important, and there's lots of different ways in which the trends in metabolomics are changing from rather more traditional metabolomics to incorporation of stable isotope work, and also the sort of, I'd almost say, rebirth of the fact that lipidomics is becoming really, really very important now. I think I've certainly enjoyed that bit. Just have a couple of slides to say that most people, when they think about a data analysis pipeline, kind of start off like that, where you've got a biological system that you're interested in, you do some measurement and some data processing. And we've heard from everyone that design of experiment is important. So here's sort of one cartoon giving that. And this is very important. I mean, obviously not many of us are working in aquatic biology. The idea here is to think that when we want to test something, that actually the only non-random variable that we want in our data or in our cohorts are the disease, if it's a disease that you're that you're looking at. And clearly that's not in that particular case, and neither is it here, where, the lady here thinks that the fish are to do with energy repair, where really they're just getting high and having a kind of great time. I'm not suggesting that we do that to our patient cohorts, or maybe the controls occasionally. But if we think of, obviously, the data analysis pipeline, then there has to be some experimental design. And I think this is obviously key. It's becoming more and more obvious in the community, but often it's quite difficult in terms of when controls are collected, how they're matched, and what the right control is. If you're looking at a disease that's to do with long-term smoking, what is an appropriate control? Is it someone who's never smoked? Is it an ex-smoker? Is it a healthy smoker? These sort of things are quite difficult for us to think about. And in that pipeline, it's also important for reporting. And the previous workshop that was next door was a lot of that was to do with how data are reported and how processing is reported. So that's important, and it's clear as a community that we should do this. and not necessarily why we did it, but how we did it, and how we did experiments, and so that we can have an ontology that allows us to have precision and understanding of our particular findings, and hopefully to have data exchange. You've heard 3 very different talks, but you'll hear lots of different aspects through this meeting, and I think what you might find in terms of clinical arenas is that there'll be a lot of overlaps in terms of what people are finding. And I bet if we did a meta-analysis in about 5 years' time, we'll finally work out what metabolites are just general inflammation markers for inflammatory diseases rather than specific markers. Yeah, okay, tryptophan. And why bother sharing data? Well, it's obviously to help your own hypothesis and maybe to use that to validate your work. We talked a little bit about numbers. David said thousands. If you're imaging, is 20 million enough? That's not a huge image, but that may be off a few people. And if we look at biopsies, is 200 enough? Is 20 patients enough? And to try to put that into context, if I can not do that, if I just click on this, I find this quite useful if it's actually going to come up, which I hope it does. Because when we look at papers, you know, we all go, oh, thousands is like really far too many to do. But that's the number of people on the planet, okay? And so if we want something that's going to have general utility, we have to think about, as David mentioned, about going into different geographic areas and different things like that. Hopefully I can get back to that. And the problem with metabolomics, as you know, is that variables are not discrete. A lot of metabolites describe the same thing, which is why the stable isotope profiling is really quite useful, because it allows one maybe to collapse things to specific pathways. Okay. We could discuss, do power calculations work? David showed some quite nice work there in terms of resampling. One assumes that you've sampled the population correctly in the start, but that's one way of doing that. And also where the variability is in terms of biology sample processing and also measurement. So those are things we could discuss. And what we want to get to, I guess, in certainly in clinical areas, and this, I can never pronounce his surname, Ioannidis? Yeah. So this is a really good paper if you've not gone into it. It's a sort of his manifesto for reproducible science. And this came out by the fact that if people have tried to reproduce papers that are in Nature and Science and Cell and things like that, it's only about 10% of them that are reproducible. And a lot of that is because of the fact that the science is not as robust as it could be, because of all of the sorts of reasons in terms of how people may abuse statistics. Okay, so that's to kick off the discussion section. We have about 20 minutes. So who would like to ask one of the panel members something? I can't use chair's prerogative. So, and that's what we're back on it. So I'm just going to move over here. So Dave showed some R squared and Q squared statistics in terms of what a PLSTA model is like. And these are used greatly in the community. Would you like to comment on their validity or otherwise, or modelling?
Oh, right. That's a tough one. Basics. I mean, people understand R squared and Q squared are. So R squared is, so when you do cross-validation, you normally split data into groups. So if you're doing 5-fold validation, we'll split it into 5 groups, build the model on 5, and then project the 5th, build the model on 4, project the 5th data set through that model, and that will give you some statistic of how well it predicts an unknown set. And then you rotate those groups and you'll get an average value of the prediction value and the modeling value. The modeling value is the R squared.
Well, actually, will this row?
No, I won't actually. If they could just sit up there, that'd be.
Testing. Okay, start again. So, if you're doing any sort of validation, even if you're doing a training test set, you build the model with a training set, you then validate it with a test set, which is projecting some unknown samples through your model and seeing how well it generalizes. So you do a prediction of some unknown samples. So your R-squared will give you a measure of how well it predicts the training data, and the Q-squared will give you an idea of how well it predicts the test set. The idea being that for it to be generalizable, they should be, really need the visual, but when the Q-squared starts dropping, that means that the model's started to overtrain. And if you're overtraining, then you fit into noise, and it will give you a false idea of how good your data is. Because the whole point of this, when we're doing this subsampling, we can't do the modeling on 10,000 samples, so we do 50, say. We want to try our best to make sure the models we produce can generalize to that 10,000. If we get really into the nitty-gritty about those 50 people, we might find something very interesting about those 50 people, but they're not generalizable to the whole population. Now, when we're doing the modelings, if you want to really need to be critical of the community, you'll often see papers where there'll be an R-squared quoted of 0.7 and a Q-squared of 0.2. And then that'll get published because that's what the software algorithm said was the point at which that inflection happens. Unfortunately, that distance, 0.2 to 0.7, means that your test set or your data set in general is not representative of the study, and that you really need to have a bigger sample size or a better set of data, because they should be very, very similar within about 0.1, 0.2, really, rule of thumb. We should really write a review on this, right, at some point. But then equally, you could say R-squared and Q-squared's a bad measure in the first place, because R-squared and Q-squared are all about difference to a predicted average, whereas you really could be using ROC curves, and so you could say the ROC curve for the training set and ROC curve for the test set, and that would probably give you a more accurate measure in terms of biomarkers. Does that answer your question?
Yeah, I think that's useful, particularly what you touched on in the middle, which was the fact that the magnitude of the R-squared and Q-squared should be similar. Because wearing an editor's hat, I quite often have people who will say, my R-squared is in the order of 0.8 and my Q-squared is positive, and therefore the model is valid, and that's actually not the case.
Yeah, there's even a kind of rule of thought somewhere happened where they seem to think that as long as something's over 0.5, it's okay. Now, if I'm reviewing a paper, I'd prefer to see an R-squared and a Q-squared of 0.4 and 0.35 than 0.9 and 0.6, because that really shows that your validation set really is representative of your training set. And it doesn't mean it's wrong. Just because you've got a low R-squared, Q-squared doesn't mean it's a bad biomarker. That's just a process of cross-validating it for the structure of your model. It's the confidence intervals and your ROC curves which will tell you actually how well it performs.
Okay, thank you. Do we have a point from the floor? Yes, please. Could you speak into the microphone? Because they're... Okay, go on then. Do you want to take that, Kevin?
Yep. So we do targeted lipidomics, so we do have a good idea on what we can see and what we can't see. So every metabolite that we're looking at, we detect at a reasonable level. So we don't run into issues where we can't correct for a batch because the metabolite's undetectable. As for reviewers, I'm not entirely sure how closely they looked at QC data. Usually we just report median CVs. We rarely give a plot like something like what I showed, and most reviewers were quite happy with it.
I think less a problem maybe with targeted analysis, which is what Kevin was saying. Maybe David can comment on targeted things.
Well, the standard protocol we put in the Nature paper is that you should be pooling from all your samples, not from just the controls. That's ideal. Sometimes that's not possible if you're doing a very, very big study. And we've been down this road with the HughesMet project, which was, we were quite naive at the time, but the only solution we had was to buy in some blood from Sigma. But unfortunately, for a lot of the metabolites, untargeted features, they weren't in the samples, you know, the samples that we bought in, the features were not in the test sample, so we had to throw away a certain amount of data. So there's now a kind of move towards having a pooled sample of all samples within a batch, and then somehow creating a lab standard, which is kind of what you alluded to, which would then put 2 or 3 in per batch across a study of multiple batches. So you correct within a batch, which is often drift, hopefully you saw it on my slide, and then you correct between the batch using that lab standard. And then you could even use that to compare different studies from different diseases, for example. Reporting RSDs is pretty standard. Doing a PCA show that your QCs have clustered really well, but remember you've got to scale your data so that you're not just overly presenting the big peaks, which have generally got a better RSD. Yeah, this is still, there's no real strict guidelines, so I think again this is certainly something that I've been speaking to Rick Dunn about, writing a paper to try and get some standardization in our report QCs. One of the most depressing things is I know labs that use QCs and then they've not made it into the paper. So I don't know how that happens, but it does happen, because I know these people are using QCs, but somewhere in the edit, whoever's leading the paper, they just don't think that it's important to report that, when it's massively important to report how reproducible your data is.
I mean, that last point's a good one. It may be that we should ask Metabolomics Workbench and Metabolites to start insisting that if QCs have been used in analyses, that they're also captured with the sample data, because they are just as important. Yes, please.
Yes, sometimes we saw very wide variation between different clinical samples. So do you usually put a reasonable cut of the value of CV within the different biological trees? How about the technology? Do you suggest another choice, the CV number?
Sorry, I missed your last point.
Okay, because sometimes we are analyzing our data, we will consider whether we would put a CV value within the biological repeats that would be acceptable, because sometimes we see quite a variation between different clinical samples.
Yep.
And also for the clinical technical repeats, whether the CV value should be lower than 10%, 50%, how about like biological repeats?
Yeah, we talked about this yesterday in the workshop. The standard is you set an RSD of 20% in your QCs, and then anything below that's okay. Obviously, the lower the better. My point of view, and I'm going to be talking about this at my Thermo talk on Wednesday, what's more important is the ratio of the biological variance to the QC variance. Personally, I don't care if I've got an RSD of 30%. If my biological variance is 200%, then that's still worth looking at. It's when your RSDs for your biology is similar to the QCs, then you know that there's no information content, so you may as well throw it away. So yeah, there's no fixed standard. It's just in the same way as when the thing for me is, and I was trying to hint at this with the targeted versus untargeted, and I was being a bit pointy just for fun. It's a stupidly valid thing to be doing. It's a fantastic thing to be doing, and as a validation step is my perspective. We do the systems biology, we see what's interesting, we see these 3-4 pathways that are interested. The next level is then to do a targeted assay where you're going to have some far more rigor and far more reproducibility. But the amount of effort that goes into developing a method for a targeted assay, somebody could take a year developing the assay with the way that they do the derivatization, the way that they do the sample prep, the way that they will optimize the techniques and do calibration curves, and all the steps that you have to go to validate. Which is great, which means you get really good data. But for the untargeted, we have a very standard method. We shove it through a pipeline, and then we just say if the RSD is less than 20, it's fine. There's no rigor at the back end about trying to map that. I know it's not directly mappable because we're doing untargeted, but that's really where we need to start looking at doing some rigorous, so that we can ensure that we only report data that's of high quality. It's not about amount, it's about I mean, it is about amount. We want to see as much as we can. But there's no point in reporting 2,000 features when that's only 100 metabolites and 70% of them, it's not reproducible. That's not getting us anywhere. So I think we really have to address that. Sorry, it's a bit of a rant, but...
Okay. Another question? Okay, I'll ask James, because you talked exclusively about 13C stable isotopes, and you kind of hinted at 15N could be used. Where would that be particularly sort of sensible to do?
Well, I mean, I think there are, again, quite good examples circulating around the TCA sort of metabolism, going into amino acids as well. We do a lot of work in the past using glutamine, which has got 2 nitrogens, and whether the label is on the amino or the amido nitrogen, you can track each one, and by knowing what you start with rather than having label on both or using carbon-13 glutamine, you can really get some intricate data and information about how the glutamine is processed. Not just in a global way, it's gone from glutamine to aspartate, for example, but precisely how it got there. And having, again, just even being able to have the amino and the amido nitrogens labeled in that way can really give you some great differences. Also, actually, even in the carbon-13 world, I talked about universally labeled glucose. You can also use glucose that's labeled in maybe just the 1 and 2 positions. And that can be very useful in looking at different activity between glycolysis and the pentose phosphate pathway, for example, because the pentose phosphate pathway and glycolysis can both feed into pyruvate, but if it goes through glycolysis, both those 1 and 2 positions will be maintained by the time they get down to pyruvate, but any carbon that's gone through the pentose phosphate pathway will have lost the first one in the first step from glucose into glucophosphoronolactone. So that can really give you some intricate details there. So it's not just simply universally labeling. It's not simply carbon-13. It's, again, getting a whole picture.
Thank you. That's useful to know. Do we have any more? Yes, please.
Yeah, that's a great question. That is experiment number 2 in any labeling experiment I do. Experiment number 1 is getting the cell numbers right. Once you've got that, every organism is different. So Marcus Rausser, who I work a lot with at the Crick, he does a lot of yeast labeling, and you can get maximal labeling in pyruvate by labeling with carbon-13 glucose after 7 seconds. And I just showed you the plasmodium results where it took 48 hours. If I was looking at lipids, I have to do 72 hours. So it's really a case-by-case basis. Every first experiment I do is choosing a number of time points, probably 0, 1, 2, 4 hours, and an overnight, by and large. Plasmodium's a bit different. And just seeing where, how long label gets into each metabolite and take it from there. Time course experiments are always experiment number 2. Yeah.
Okay, any more? Yes, please.
Hi, this is for the first speaker. You put a lot of attention on sample size to get your resources as generalized as possible. But research is going towards more like a personalized, especially in medicine. What do you think after recent months, should we get more important to smaller samples?
Yeah, I'm glad you asked that, because really the only thing I'm really interested in terms of future work is longitudinal studies. Time is everything. It's knowing how personal trajectories, how disease progression, how response to an insult, exercise, diet, single time point, how interesting is that? It's not. We have the ability with metabolomics to measure things really, really quickly, so we should be using that. For the very, very personalised, the N of 1, that's a tricky one. I mean, that's something we all discuss about. I can only think that we need to start having ways of collecting blood, maybe blood spot, so that you could measure basically yourself a lot, so that you become your own baseline. So if you're measuring yourself week after week after week, and then one week something changes, well, you've got a lot of data there that can help with the power of the statistics, because you've got that baseline really well defined. And assuming that the effect size is big enough, this is always going to be the issue with statistics, then that will help. If there's something that's happened regularly, then that's going to get even more powerful. So using yourself as a control and people monitoring themselves all the time throughout their lives is where we're probably going to end up doing. How we do that is a different matter, but I think that's where it's going. The other area that's particularly interesting, and Roy could probably speak more about this, is on the imaging side of things. By having having the data in the resolution rather than the single sample, so you can get away with having, say, 20 samples, but if each one is an image of 20 million pixels, then you can start looking at the three-dimensional dynamics of that. That's another way of, you're not recruiting as many patients, but you're trying to get as much data out of them as possible. And I suppose the other way is multiple systems. If you can measure blood, urine, microbiome, the more systems, the more you combine that data, the more powerful your modeling is going to be.
So some of that's starting to happen. So there's some nice electrochemistry now that will measure glucose with patches that are embedded under the skin that also feed back to insulin pumps and things like that. The issue is discovering what the next marker is to do that. And as David indicated, I think there has to be quite a different shift in how samples are collected, because his point is collecting samples when someone's already ill. Because if there's a nice plot that Jan van der Graaf published, well, it could be 15 years ago now, and it was sort of the life history of someone as they started off in a sort of area where they're quite healthy and then they get unhealthy. And the key thing to do is to find the disposition or the predisposition markers. But that needs really quite a different strategy in terms of healthcare, I think, which would be really interesting, and it would be nice to see that start, actually. But I think ethically quite challenging to get that through review panels. Okay, I suspect we've dried up, so that's the end of our discussion. I'd like to thank our three speakers for their nice talks, and also you for your nice interaction with them and for the discussion. Thanks very much.
Thanks, Roy.
Rapturous applause.
Hello.